Está bonito porque empezamos con carreras de bioquímica no son muchos, son poquitos alumnos. Es que se está integrando Texas University. Están ahorita poquitos alumnos de bioquímica, pero hay. Gracias. Entonces, por ahí a lo mejor si vienen ahorita nos representan.
La sección de preguntas
para más práctica. Sobre todo, yo estoy dando ya está bien. Listo. Ok. Okay, guys, welcome. We continue with our talks. The topic. Just an announcement. The next talk after this one, unfortunately, it's being cancelled. The speaker had a problem, medical problem with her husband, and unfortunately, it has to be cancelled. So this will be the last talk for today. And if you have questions at the end, remember you can ask the questions in Spanish or in English if you feel comfortable. But it can be clear.
Okay, so hello everyone. So the next stop is biochemistry, applications in health and sports, which will be given by Dr. Luis. He is a biological chemist based in Mexico, specializing in functional and orthomolecular medicine. His work focuses on chronic disease, aging, and the regulatory systems of the human body from a biochemical and integrative perspective. He has completed advanced training in functional medicine, orthomolecular nutrition, endocannabinoidology, and Japanese acupuncture, and serves as an instructor and master trainer for healthcare professionals. He collaborates with organizations such as the Asociación Mexicana de Educación Deportiva and the Asociación Mexicana de Psiconeuroinmunoendocrinología. He is a frequent invited speaker at national and international scientific events, where he translates complex biochemical concepts into clinically practical and educational rigorous frameworks. So let's give a big round of applause for Arturo Guizar.
No sé si está bien. Lo chequé con sus instancias y okay, good afternoon. My name is Arturo Guizar Castro. I'm a specialist in functional nutrition and integrative medicine. Today's presentation will verse about biochemistry, especially its applications in sports and health. Okay, as we said, in order to keep the timing precise, I will let questions and answers at the end. Of course, we can make it in Spanish. So it will be, I hope, a very interesting lecture. Let's start. By start, let's make a brief interview about biochemistry. You can hear me? Yes? Everybody? Everybody? Okay, then we start. Okay. Biochemistry is a scientific discipline that explores the chemical and molecular processes occurring within living organisms. It serves as a foundational science for understanding life at its most fundamental level. By examining the interactions between biomolecules such as proteins, nucleic acids, lipids, and carbohydrates, biochemistry provides insights into cellular functions, metabolic pathways, and physiological mechanisms. This field bridges biology and chemistry, enabling researchers to decipher the molecular basis of health and disease. Its applications are vast, ranging from clinical diagnostics and therapeutic development to enhancing athletic performance and recovery. As a result, biochemistry plays a critical role in advanced medical science, nutrition, pharmacology, and sports science. The integration of biochemistry with emerging fields like molecular biology, genetics, and biotechnology has further expanded its relevance, making it indispensable in both research and applied health sciences. Let's make a brief review about the history of biochemistry. Okay? The history of biochemistry traces back to ancient philosophical inquiries into the nature of life. Early thinkers like Aristotle and Galen speculated about vital fluids and the essence of life. In the 17th century, Van Helmont conducted pioneering studies on fermentation and digestion, laying the groundwork for biochemical chemistry. Emergence of organic chemistry marked a significant milestone, with Lavoisier linking respiration to oxidation and Waller synthesizing urea from inorganic substances in 1828, challenging the belief that organic compounds could only arise from living organisms. The 19th century saw key discoveries such as Payen identifying the first enzyme diastase and Buchner demonstrating that fermentation could occur without living cells. Liebig contributed in a chemical theory of metabolism, further solidifying this field. The term biochemistry was formally adopted in late 19th century, with Carl Neuberg recognizing as a foundational figure. The 20th century brought transformative advances, including the discovery of vitamins, hormones, and coenzymes, the identification of DNA as genetic material by Avery in 1944, the elucidation of the DNA double helix by Franklin, Watson, and Crick in 1953, and the deciphering of the genetic code of the 1960s. Contemporary biochemistry now integrates with molecular biology, genetics, biotechnology, and personalized medicine, focusing on areas such as metabolic pathways, cellular signaling, and epigenetics. Talking about applications of biochemistry in sports. It will be a brief presentation because it's vast. It's the subject for a diploma, even a full course. Well, in the realm of sports, biochemistry provides critical insights into optimizing physical performance, preventing injuries, and accelerating recovery. One of the primary areas of focus is energy metabolism, where biochemists analyze how ATP is produced from carbohydrates, fats, and proteins. This knowledge informs nutritional strategies tailored to the specific energy demands on different sports disciplines. Biochemistry also examines acid-base balance and muscle fatigue by studying the accumulation of lactic acid and other metabolites, which support the development of effective recovery protocols and training methodologies. Performance and recovery biomarkers such as lactate, creatine, kinase, cortisol, and testosterone are monitored to assess physical status and prevent overtraining. Functional sports nutrition applies biochemical principles to enhance protein synthesis, cellular repair, and hormonal equilibrium, often incorporating evidence-based supplementation. Furthermore, the genetics and epigenetics of performance are explored to understand how genetics variants influence training responses, enabling the personalization of athletic programs. Anti-doping control is another crucial application where biochemical assays are used to detect prohibited substances, ensuring fairness and integrity in professional sports. Let's talk a little bit about metabolism. I'm pretty sure you already know a little bit about this, or a lot, I don't know, we'll see later. But well, it's good to make a brief review. Metabolism is the set of chemical reactions that occur in cells to obtain energy. It regulates processes such as digestion and protein synthesis. And efficient metabolism is key to maintaining body balance, influencing weight, and the body's overall well-being. It can be divided in anabolism, which is a metabolic process that builds up complex molecules from simpler ones using energy, or catabolism, which breaks down complex molecules into simpler ones, releasing energy. Both processes are essential for energy balance and proper functioning of the body. Another important concept for metabolism will be basal metabolism, which is the minimum energy that the body needs at rest to maintain vital functions. And total metabolism includes basal metabolism plus the energy required for daily activities and digestion. Both concepts, basal and total metabolism, are key to understand energy expenditure and body weight control. It's what is measured by calories. Basal metabolism can be regulated, and there are some factors that can activate the basal metabolism, which can be the muscle mass that increases basal metabolism because muscles consume more energy even at rest. Why? Because they have an increased number of mitochondria. Regular physical activity stimulates calorie burning and improves metabolic efficiency. In addition, factors such as age, gender, and thyroid hormones significantly influence the basal metabolic pair. Talking about mitochondria, mitochondria are the cellular organelles that generate most of the chemical energy needed to activate the cell's biochemical reactions. This chemical energy produced is stored in a molecule that you already know as ATP, adenosine triphosphate. The number of mitochondria varies from less than 100 to several thousand, depending on the amount of energy each cell and tissue requires. That is flexibility, the flexibility of mitochondria. Mitochondria also has a dynamic character that allows it to vary size and shape, and it can be divided, fission, fused, or deformed if necessary, and even can be recycled in case of an aging mitochondria. All the materials can be recycled. That's flexibility. Some mitochondria, are only a few hundred nanometers in diameter and globular in shape, while others are elongate or filamentous or even branched. As you can see in the diagram, you can see the fission of several mitochondria to form a clover, as we see, or even a mitochondria that is aging at the right, and that's recycled by autophagia, mitophagia. Mitochondria have also the ability to reproduce on their own. This is because it has its own DNA that is called mitochondrial DNA, which allows for more mitochondria as the cell needs to obtain ATP, even more ATP. That's what it's called mitochondrial flexibility. Mitochondria generate cellular energy by producing ATP through respiration. I think you already know. By mitochondrial uncoupling protein called UCP1 can generate heat that will maintain and regulate the temperature in the body. It also regulates cellular processes such as apoptosis, helping to maintain healthy cellular balance. In addition, it participates in the metabolism of nutrients and the synthesis of certain lipids and proteins essential for cell function. In the slide at the left, you can see where the UCP protein is located. At the left, you see the electronic chain of electrons, and at the end, you see the ATPase. We'll talk about that later. But in the middle, there is a protein, UCP1, that uses the electrons from the electronic chain to produce heat. Why is so important? Because, as you already know in chemistry, there is a thermodynamics law that is called Q10. I don't know if you're familiar with that law of thermodynamics. And it says that in a given circumstance, a biological reaction, or even any reaction, increases the speed in terms of 10 degrees intervals. That's why in a laboratory you have a Buchner, you have stoves, you have a nest, you have things to heat up the reaction. That is because if the temperature increases, the energy of the reaction increases too, and then makes it twice faster by each change in 10 degrees centigrade. That's called the Q10 law. In the body, this change is not measured by degrees, complete degrees centigrade. It's measured by cents or decimals from a degree in order to accelerate these reactions. In this case, the body maintains a steady temperature to maintain the body reactions. That's why we need the uncoupling protein, okay? And it's situated at the end of the electronic chain transport. Okay. I'm talking about energy systems. I think you already, I'm pretty sure you already know much of this. Then after this review, we'll enter into our matter. This is like a brief introduction. Energy systems, the energy necessary for the functioning of the body is obtained primarily from the metabolism of carbohydrates converted into glucose. You already know that. It can also be obtained from the metabolism of essential fatty acids or amino acids. The third way to obtain energy is called a lactose anaerobic pathway or phosphagen pathway. And utilizes phosphocreatine as a molecule similar to ATP, which is stored in muscle cells. The phosphagen pathway is crucial for rapid muscle energy during intense and brief efforts, such as sprints. It uses phosphocreatine to regenerate ATP, allowing bursts of force and speed. This system predominates in activities such as weightlifting or short running or even swimming, that is called sprints. I think you are familiar with sprints in the sport. Well, this effort uses phosphocreatine as a principal source of energy. The system predominates in activities of short duration and intense intensity. Why? Because this is called like instant energy. As we already know, we have hybrid cars that uses two motors, one electric, one of combustion. In most of the hybrid motors, if you start moving, the principal motor that functions, it's electric. That's why it's so silent. Then as speed increases, then the combustion motor integrates and gives you extra power. In this case, phosphocreatine would be your electric motor starting to move. Then to keep a steady pace, then you start the glycolysis, the beta oxidation, and the other pathways that continues with the energy in a more steady way. Is it understood? Is it clear? More or less? Okay, let's continue then. Cellular respiration is critical for the production of energy cells. It consists of the breakdown of glucose and oxygen to generate ATP, the energy molecule. This process occurs in the mitochondria and are divided in three stages: glycolysis, Krebs cycle, and oxidative phosphorylation. Of course, you already have exams about that, right? Yes? No? Yeah? Okay. Will not be so that will be easier for us. We will verse about how can you apply this knowledge in daily basis. Okay? Okay. Glycolysis is the anaerobic degradation without oxygen present of glucose into pyruvic acid, right? No. None of the above? Okay. When the demand of energy from ATP is excessive, pyruvic acid is transiently converted into lactic acid. If you already have made any effort like gym or any sports, after the exercise, you have some muscle pain, right? Well, that's because of the lactic acid, okay? Which provokes this pain, this tiredness is called early fatigue, and it's because of the accumulation of lactic acid. The total gain of this metabolic pathway is 32 ATP molecules per glucose molecule. That is 15 ATP for each molecule of acetyl coenzyme A, 15. And 2, which are 2, that give you 30 ATP, plus 2 ATP by the process of obtaining the pyruvate molecules. Okay. Beta oxidation, on the other hand, is the main catabolic process in the mitochondria for fatty acid degradation and energy production. It takes place mainly in the mitochondrial matrix, although it can occur in peroxisomes also. This mechanism comprises a series of 4 reactions that successfully eliminate 2 carbon fragments in the form of acetyl coenzyme A, also generating NTH and FDH2, which are essential during periods of fasting or physical activity. The energy gain from beta oxidation can be up to 129 ATP molecules per fatty acid of about 16 carbon atoms. As you can see, it's even more efficient than glycolysis. That's why when you start in an exercise and you get into rhythm, or it's a very long exercise, you can start feeling not so tired, and you can continue the second breath, as some people talk. That's because beta oxidation gets into the place and it starts giving you extra energy. Your favorite? No. Blood cycle? Not the favorite? Beta oxidation maybe? No. Glycolysis? No. Anybody? No. No. Mine is Krebs and phosphorylation. The layer. The purpose of Krebs cycle is to obtain NDH and FDH2, which already have speak about. These molecules have reducing power and are essential for the production of ATP. It occurs in the mitochondria as part of the aerobic respiration, and it's carried out in the presence of oxygen and develops the process of between the processes of glycolysis and beta oxidation and the respiratory chain. It also directs excess energy. I know my English is not the best. It's very Mexican. I know. It also directs excess energy towards the biosynthesis of fatty acid, which allows energy storage. My favorite. Oxidative phosphorylation comprises the electronic chain of transport of chain and chemiosmosis. In this step, NADH and FDH2 obtained from previous phases like Krebs cycle or glycolysis or beta oxidation. All these molecules get together into the chain of transport and then will be transformed into ATP thanks to the movement of electrons through a series of proteins embedded in the inner membrane of the mitochondria. At the left of the slide, you can see the electron transport chain. You already remember that? Yes. I'm impressed. Okay. The electron transport chain occurs in the mitochondria matrix, okay, during cellular respiration. If you see the left of the slide, the bottom will be the matrix of the mitochondria. The upper membrane that you see will be the outer the membrane of the mitochondria. And in the middle, we call it intermembrane space. Okay. We use in this chain the matrix down and the intermembrane space. Okay. It consists in a series of proteins that transfer electrons, generating a proton gradient. You can see at the bottom 4 protein complexes. I don't know if you can see it, but they are numbered in Roman numbers. 1, 2, 3, 4. Every time NDH or FDH participate with this, interact with these proteins, electrons are liberated. You see it in the yellow circles, and they liberate protons, which are the molecules that are the hydrogen with a positive charge. They accumulate in the intermembrane space. And this functions like a battery. It generates a gradient, and then at the end of this chain, you can see a molecule called ATP synthase. It's in the right in the extreme, okay? This functions a little bit like, I can help, like a generator. If you already saw, like it's called a dynamo that uses water going down through a tunnel and moves a generator. This starts spinning and you can obtain electricity. In this case, what you obtain will be ATP. Okay. During a workout, the use of energy system varies depending on the duration and the intensity of the activity. At the beginning, the phosphagen system predominates due to explosive stress. As time increases, the glycolytic system becomes active for short to medium duration activities. Finally, the prolonged workouts, the oxidative system is the main provider of energy. When this happens, as you can see, after a long period of exercise, fatigue comes around. Okay? Fatigue significantly limits physical performance due to several triggers. First, increased acidosis of lactic acid builds up into the muscle, and that impairs the muscle function. Second, depletion of glycogen and glucose. That reduces energy ability and for sustained activity. And this is important. Why? Most people can tell you, okay, I'm finishing my reserve of glucose or glycogen. Okay, I can continue using my fatty acids. That is not true. In order to start beta oxidation, you need a reserve of carbohydrates. If you do not have any glucose left, then you cannot start the beta oxidation. You cannot use your fatty acids as a source of energy. That is so important. Then, during a training, you need to ingest carbohydrates, glucose, in order to continue the exercise and get the ability to burn your fatty acids. That is something not always taken into account, but it's basic, okay? Third, neuromuscular transmission can fail due to decreased amino acid impacting neurotransmitter synthesis and muscle activation. If you lack of amino acids, you cannot synthesize neurotransmitters. If you do not have neurotransmitters, you cannot order to your muscles to move or to continue moving. Okay, then you need the ingestion of proteins before an exercise and even during or at the end of an exercise. Okay. Also, during fatigue, you can observe the increase of the concentration of an amino acid called tryptophan that converts into serotonin. You are familiar with the neurotransmitter serotonin? Yes? Serotonin is a neurotransmitter responsible with GABA of make you sleepy, like right now. I know my voice, it's so soft, it's magnificent. And if you have classes after lunch, it's necessary that you get sleepy. That's the accumulation of tryptophan converting into serotonin. How can you manage this? There are some amino acids, you must know, even people that go to the gym knows them. They are called branch chain amino acids, BECAs. You're familiar? BECAs, BCAA. Those amino acids help you to diminish the concentration of tryptophan, serotonin into the brain. That gets you awareness. Then you need proteins. Well, there is a difference between overtraining and fatigue. Now we're talking about sports, but we can translate this into the ordinary life. Let's see about that. If you are tired normally, then you feel sleepy, you feel lazy, I don't know. In the state of tiredness, you can recover quickly by resting, sleeping, or making any other thing, not sports, not activity. Okay. In the state of tiredness, we can recover quickly through rest and good nutrition. We already talked about amino acids, but we already talked about carbohydrates. If you are overtrained, if you are too tired, you need longer periods of resting. Okay. When stress overtakes resting for several days, you begin to feel fatigue and exhaustion relentlessly, constantly. That is called fatigue, not just overtraining, not just tired. Okay? The body's ability to adapt is affected, and the protective processes are activated to prevent further damage. Makes you sleepy, your immune system goes down, that you can get sick easily, or you cannot concentrate, or I don't know, depressed. Okay? Well, that is called chronic fatigue. The body is turning down all your activities, neural activities, muscular. The body is start to make in a standby basis, and we say that, in order to protect you, to avoid, how can we say it? To get stuck, to get too tired, even to be alive. I don't know if we can say it like that. But the body it starts to protect itself, so you cannot break down. That would be okay.
Quick interruption. I actually figured out, yeah, there's an issue with the session already. Yeah, touch something quickly.
Ya no dormí. Okay. Now get busy. So far, we have talked about biochemistry, as you already know, like the books. A little interruption. Now let's talk about doping. What about doping? Okay. Doping consists in the use of prohibited substances to improve sports performance. This practice puts the health of the athletes at risk and is sanctioned by international organizations. In addition, doping affects the integrity and fairness of sports competitors and generating controversy and mistrust. I know you already have heard the term doping. Okay. What can be a definition of doping? It's the use of an artifice, substance, or method potentially dangerous to the health of athletes and or likely to enhance their performance, or the presence in the athlete's body of a substance or the verification of a method which is listed in the anti-doping code of the Olympic movement. At the left of the slide, you can see a screenshot from the World Anti-Doping Agency, the WADA. This is the organism, international organism, that prepares a list of prohibited substances called doping, but not only substances, stimulants, narcotics, anabolics, diuretics, whatever. Methods, blood doping, we can talk about that in the Q&A. Pharmacological doping, manipulation, chemical handling, or anything else, or circumstances. I was talking about marijuana, for example. In general matter, marijuana is not allowed in most of the sports, but in some circumstances, the WADA can allow an athlete to consume it in a therapeutic way after a lesion, an injury, or any other circumstances. And there are sports where this is totally prohibited. Shooting, for example, driving. Okay, in those cases, it's completely forbidden. But there are some sports that can allow in certain circumstances. Okay, those are the list of the WADA permits, and it's refreshed every year. Every year it gets renewed. There are many substances illegal in sports. The most known is testosterone, as most of us already know, because of the abuse of steroids during build-up muscle. A number of methods have been employed to detect testosterone in athletes, mostly based in urine tests. Let's see about that. This is more for the chemists. This is called the testosterone epitestosterone ratio. It's not the same molecule, but almost. At the left, you see a molecule that is called testosterone, the natural testosterone. And at the right, you can see a molecule that is called epitestosterone or episterone. It's the same. It's almost the same. Almost, not quite. The oxidryl group, the OH, at carbon 17 is forward facing at the right. When you see those that bold line, that means the oxidryl group is going towards you, facing you. And at the left, testosterone, the oxidryl group is going backwards. Okay? This is like a fluttering. Okay? That occurs only in natural testosterone. Synthetic testosterone cannot do that. Okay? Then what we do? We take a sample of urine and we measure the quantity of testosterone and epitestosterone or episterone. And we make a ratio. If the TA, the testosterone episterone ratio goes from 1 to 4, that means most of the testosterone is natural, comes from the body. So it's fair. If the TA ratio builds up beyond 4, 5, 6, or I don't know, that means that most of the testosterone of this athlete is synthetic. Then it's a doping case. Okay? That's one. Let's see another. If you're familiar, the LH is the luteinizing hormone. Testes produce testosterone, ovaries produce estrogen, okay? And that is under the feedback control of hypothalamic pituitary axis, okay? That is the figure at the right of the slide, okay? The feedback about this is like that, goes like this. If there is a high concentration of LH, luteinizing hormone, that means that you have low concentration of testosterone. That means the testosterone is feedback and there is no much synthesis of testosterone. High LH, down testosterone. On the contrary, high concentration of testosterone means you have provokes a lower concentration of LH. That must be inverse. Okay. Then you get the competitor, the winner, or they take the fifth, they take the five first, but concentrate mostly in the first three. They take urine samples and they measure. They measure first testosterone, then they measure LH. They must be inverse. If by any circumstances you find high testosterone and high concentration of LH, that means that this person is taking recombinating luteinizing hormone. That cannot be in a natural way. That means doping. Okay? Yes, it understands? Okay. These are things that cannot not everybody discuss and are from every day in sports. Another one, more chemical, even more chemical, the C13, C12 ratio. Carbon has 3 different isotopes, 13, 12, and 14, okay? Carbon 12 is the most abundant. Pharmaceutical companies typically synthesize testosterone from molecules from plants in the same family as soybean. Okay? Soybean comes from a hot climate, okay, and have a lower proportion of carbon-13. Our diet is rich in carbon-13 because our climate is not so hot. Then what you eat with corn, I don't know, oats, that means those foods, those grains, are lower in carbon-12 and higher in carbon-13. Then, you take the sample, you measure all the hormones in this competitor, and you determine the concentration of carbon-13. If the composition of the proportion of carbon-13 in testosterone is lower than the other hormones, that means this person, this competitor, is injecting or taking, by the way, an artificial form of testosterone because it doesn't resemble to the composition of the other molecules. Well, that's about sports. What about health? Let's see. Biochemistry plays a pivotal role in modern healthcare by enabling the understanding, diagnosis, and treatment of various diseases. In clinical diagnostics, biochemical analyses are essential for detecting abnormalities in metabolism, hormone levels, and enzyme activity. Common tests include glucose level, lipid profiles, liver enzyme, electrolyte panels. Functional, by area, and clinical nutrition relies on biochemistry to understand how nutrients interact with metabolic pathways, allowing for personalized dietary interventions to manage chronic conditions. In pharmacology, biochemistry helps elucidate how drugs are absorbed, metabolized, and excreted, optimizing dosage and minimizing adverse events. Molecular genetics and personalized medicine use biochemical techniques to identify genetic variants that influence health outcomes, paving the way for targeted therapies and individualized prevention strategies. Biochemistry also addresses chronic and metabolic diseases such as diabetes, hypothyroidism, dyslipidemia, and mitochondrial disorders by developing biomarkers and therapeutic approaches. Also my area. In cancer and neurodegenerative research, it investigates disruption in cellular signaling, apoptosis, and oxidative stress, contributing to innovative treatments. Additionally, hormonal and reproductive health assessment depend on biochemical evaluations to maintain physiological balance and support overall well-being. What's the difference between aging and senescence? Are the same? No, not really. Aging can be defined as the breakdown of cell organization system and the reduced ability to adapt to the environment. Senescence is the progressive deterioration of bodily functions over time, and you lose your ability to adapt. Cellular senescence occurs when cells stop dividing, contributing to aging and age-related diseases. Senescence is the process of cellular aging and leads to loss of function. In addition, senescence is related to chronic diseases and physical deterioration, being key for understanding aging and developing anti-aging therapies, also my area. Research into aging explores genetic, environmental, and lifestyle factors to better understand how to promote healthy longevity and mitigate age-associated decline. Serotonin is increasing. Once again, in cellular respiration, electrons exchanged through the electronic transport chain invariably escape and leak into the cell's respiratory centers, generating toxic reactive oxygen species. Maybe you are familiar with the term free radicals. That's more likely. Well, a free radical, as you see in this slide, you see an impaired electron in the outer orbital of an atom. That makes unstable the molecule, but the atom. It needs another electron that has to take anywhere in order to stabilize. That is the mechanism of we are called free radicals reaction. That's what is happening with the other molecule, as you can see. Okay. One atom receives the electron, it gets reduced, and the other loses the electron, gets oxidized. That's what it comes. You are familiar with the term redox. That's it. Okay. Examples of the reactive oxygen species are in this slide, and they can cause oxidative damage in the cells. This damage promotes cellular senescence, and the process is the process which cells lose their ability to reproduce. The accumulation of senescent cells contribute to aging and related diseases, such as cancer and degenerative disorders. The first line of defense will be the free radicals, the endogenous enzymes, okay? These are enzymes made by the own cell. They work together to get rid of the toxic oxygen derived free radicals generated by respiration. Antioxidants at the left of this slide neutralize reactive oxygen species, ROS, preventing cell damage, okay? And they help to maintain the redox balance, donating electrons, as we see at the right. You already saw that part of the slide. Protective lipid proteins and DNA. But as you can see, they are not only enzymes like catalase, glutathione peroxidase. At the bottom, you can see in the yellow underline vitamins, vitamin A, vitamin C, flavonoids, carotenoids, and glutathione. I don't know about you, but when I was studying biochemistry at the university, and they started talking about vitamins, I say, vitamins, nutrition, minerals. Yeah, the most boring thing, right? Well, if I can tell you that you can save lives with this, that you can stop cancer in some cases, would be interesting for you? Well, that's my area. That's what I do. That's part of what I do. I also help some sports, some competitors, Olympic level, to get to their goals. That's when you start to feel, okay, biochemistry can be of any use. Maybe. As we age, we slowly lose our ability to manufacture these enzymes. Free radicals accumulate, and then oxidative stress and aging gets accelerated. Okay? 2009 Nobel Prize in Physiology or Medicine was awarded to Elizabeth Blackburn, Carol Greider, and Jacques Sostak. You are familiar with them? Yeah? Okay. They discovered how chromosomes are protected by telomeres. You are familiar with telomeres? Okay. And the function of the enzyme telomerase. You are familiar with the enzyme telomerase? Yeah? Okay. That's the subject of the 2009 Nobel Prize. In a brief description. Cell reproduction allows tissue for growth and regeneration. Telomeres in the chromosomes are repeated sequences of DNA that protect the ends of chromosomes and prevent their deterioration, what you see at the left of the slide. During cell division, telomeres progressively shorten, right? They get short every time a cell divides because of the copies of the DNA. Well, telomere shortening is associated with cellular aging and limits the number of divisions that a cell can make. It's more like the license you have for the Windows program. You have permission to make some copies, 5, 4, I don't know, depends on the license. Once you finish all the license you have for the program, the Windows, you can make another copy? No? Well, same here. When the cells reaches the end of the permits, then there's a signaling that gives you that leads you to apoptosis, programmed death of the cell. Okay? That happens only if telomerase enzyme is not active. That is in most of the tissues. But if this enzyme is active, telomerase extends the lifespan of cells by adding repetitive sequences of DNA to telomerase, as we see at the right. Its activity is related to cellular aging. When telomerase is not active, then the cells get a limited number of reproductions. But if telomerase is active, then you regenerate the telomere, and the cell can divide enzyme another after another after another after another with no limit. That's the mechanism that cancer cells use, the active telomerase. Okay. That, and you can add it with the free radicals provoking senescence. Okay. When telomeres make the signaling that you can no longer divide, there it activates the P53, the P53 protein by the telomere dysfunction, and this protein says like this, makes like this. P53 protein makes a stop. You want to divide the cell, but there is an error or whatever, or even the permits are finished. Then P53 protein stops the process, okay, and gives you time to another proteins like the DNA polymerase or any other to rebuild the damaged part of the DNA. Okay. If the reconstruction is successful, then the cell can divide again, then continue the division. Okay. If not, then that calls for the apoptosis process. Okay. Then P53 protein, it's like the gatekeeper from cell division. You can see that. Okay. We already talked about this. When P53 proteins tells you that you can no longer divide, then apoptose, the program cell, the cell, its program, and it can be triggered also by the loss of function of the telomerase of the P53, or maybe if an infection, lesion, damage, mechanical damage, toxic agents, or lack of nutrients. Denutrition is one of the triggers for apoptosis. Okay, and not everybody is aware of that. Well, how can we manage this part? They say that malnutrition can unlock these kind of processes. Are you familiar with the term epigenetics? Yes. Oh, very smart. That will make very interesting the Q&A. Okay. Epigenetics studies how environmental factors and lifestyle can modify gene expression without changing DNA sequences, right? No mutations. Okay. It explains how identical genes can lead to different results due to chemical markers added to DNA. Understanding epigenetics offers insights into diseases development and potential therapeutic approaches or personalized medicine, also my area of practice. Well, you remember chromatin? Yes? You already had exam about this, right? That's why it's so fresh. Yes? Then let's review. Chromatin is composed of DNA and proteins that generate compact structure, as you can see at the left. Crucial for packaging and the stability of chromosomes, right? Okay. The main proteins are called histones, right? Okay. These histones, it's when their DNA is coiled to form a nucleosome that you see at the left. that's a nucleosome. The epigenetics involves covalent chromatin modifications that you see at the right and do not affect the DNA. There is no mutation, right? DNA, it's intact, doesn't move, it's not really a system situation. These covalent modifications affect both chromatin structure and the recruitment of transcription complexes. That in effect turn genes on or off. There are two modifications that can take place, as you can remember, acetylation and the other one, methylation. That makes changes in the histones and then permits or not the transcription or not of certain genes. What that happens, that makes changes in the manifestation, the physical manifestation of certain processes. Okay. These dynamic epigenetic modifications are carried out by adding, writing, and removing, deleting post-transactional modifications, followed by the reading, which determines the genetic expression and phenotypic response. Okay. How does epigenetic can apply in everyday life? Well, the less known of these situations can be fetal programming. You are familiar with this? No, not much, just really. The rest, not really? Okay, let's talk about this. Pregnancy is a stage of great inflammation for women, okay? Due to the implantation and growth of the embryo, the fetus absorbs 40% of mother's anti-inflammatory reserves, amino acids, omega 3, 6, and 9 fatty acids, vitamins, minerals. No. The fetus takes it all, and you have nothing to do about it, okay? You cannot do anything about it. Replacing this deficiency that pregnancy brings up could require at least 4 years for the mother, 4 years for the mother with a balanced diet, a perfect balanced diet. And with this, the mother needs 4 years to recover from each pregnancy process. Okay, what happens if this doesn't achieve? Well, when pregnancy has a high burden of inflammation and or malnutrition, pro-inflammatory genes are activated in the fetus that under certain nutritional conditions can trigger chronic diseases in the future. What happens? The fetal feels, how can I say it? The environment is aggressive, very aggressive for the fetus, and the fetus has to defend itself. And you have a huge library of genes available for you as a fetus. Then the fetus can select which genes activate or deactivate to protect itself against the environment or the lack of nutrients. It tries to overcome the situation. And that provokes epigenetic changes in this DNA that gets in a latent state, not expressed, until you get born, and environmental situation triggers these genes. Okay. Then malnutrition increases maternal and infant mortality or morbidity. A child with low weight at birth can be increased. The risk of morbidity or mortality in the medium and long time will be present, depends on the circumstances that can trigger this. But the most important thing, malnutrition or epigenetic adaptation among women and men, not only the mother, okay, increases the likelihood of reproducing these altered patterns into the next generation. That means if you are malnourished, you activate these genes, not expressed yet. That happens in the future. You carry these adaptations and you transmit this into your child. That means that what you do here can affect even to your grandchildren. If you do it right, it will be good for your grandchildren. If you are doing something not right, it will transfer even into your grandchildren. Okay, that's why it's so important. Well, what we do now affects our future generations. Can be the reasoning, the conclusion for this. What about cellular inflammation? We already said that pregnancy is a state of high inflammation for the mother. What is this? Well, the interest in participation of lipids in this process began when they studied thank you, the effect of the body's immune responses. Fatty acids are not only energy storage. Okay? These fatty acids have special functions in our body, and that's what we started to study last century, 1 century ago, but gets more important even these days. We'll speak about that. You already know about inflammation. No? What inflammation is? Well, inflammation is a reaction of the body that helps to defend itself against aggressions that damage the organism. The brain orders the production of eicosanoids. Eicosanoids, you know what eicosanoids are? No? Okay. The brain orders production of eicosanoids and in the affected area or body to limit damage and starts the healing process. There are two kinds of inflammation. Acute, it hurts. What hurts? It's a rapid and short-lived response to injury, infection, characterized by redness and pain, what we already know. Silent inflammation, it's also called chronic inflammation, and persists over time. It doesn't hurt much. If you feel it hurts, what you take an analgesic, and then you get rid of the problem. But if it doesn't hurt, what do? Nothing, because you are not aware. Then chronic inflammation persists over time, and it can damage tissues and cause diseases in the long time. Okay. Chronic inflammation can be triggered by persistent infections such as bacterial virus that the body doesn't completely eliminate, stress, a diet high in starches, refined sugar, saturated fats also contribute to the inflammatory process. In addition, autoimmune diseases cause prolonged immune system responses, keeping inflammation active. Chronic inflammation can contribute to the development of diseases such as diabetes, cancer, and cardiovascular problems. Any familiar? Rings any bell? Yep. In addition, it affects the immune system, weakening the body's response to infections or allergies. It triggers allergies. We are about to enter a season of allergies. Spring. Okay. Finally, it generates sustained tissue damage, which can cause persistent pain and decrease quality of life. Eicosanoids are lipids derived from fatty acids. They come from the number 20, eikos. That means 20 Greek. Eicosanoids, fatty acids of 20 atoms of carbon or even more. Mostly 20 atoms of carbon, eikos. They modulate the activity of immune cells such as macrophages, lymphocytes. In addition, they participate in the intercellular communication to control processes such as fever, pain, vasodilation, or even infections or injuries. Eicosanoids were the first hormones produced by living organisms. They are divided by their function in two main categories: pro-inflammatory eicosanoids, anti-inflammatory eicosanoids. Pro-inflammatory eicosanoids promote platelet aggregation. They are vasoconstrictors. They promote cell proliferation, and they alter the immune system. On the contrary, anti-inflammatory inhibit platelet aggregation. They are vasodilators. They regulate cellular proliferation and the immune system. Inflammatory eicosanoids can destroy tissues, and they come from omega-6 fatty acids, such as arachidonic acid. You are familiar with arachidonic or the omega-6? Omega-6? No? Maybe the other. Anti-inflammatory eicosanoids promote healing. They come from omega-3, omega-3. Then you are familiar. Like eicosapentaenoic acid. If you see these fish oil capsules and you see the label, you can read APA, DHA. Those are omega-3. Okay? The diet is rich in omega-6, arachidonic, our diet. That comes from diet. We can supplement APA, DHA by fish oil capsules. These two types of eicosanoids must coexist in a balanced way to keep us healthy. As you see at the right of the slide, these omega are stored in the phospholipids of the cell's membrane until they are needed in the upper part. If necessary, they can be liberated from the cell's membrane by a phospholipase enzyme, and as free fatty acids can be transformed into the corresponding eicosanoids that are the molecules that you see at the bottom. Those are eicosanoids, can be pro-inflammatory or anti-inflammatory. It depends on the omega-6 inflammatory or the omega-3 anti-inflammatory. Diet is the simplest and most effective way to achieve a balance between both types of eicosanoids. That's my area. Modern diets, we usually has a 10-to-1 ratio of omega-6 to omega-1, even going to 30-to-1. That's the most common diet. Even 30, most of the diet actually comes to 20 to 30-to-1. The suggested ratio for healthy persons can be 4-to-1, not 30, 4-to-1. Then we realize that we are submerged in a very inflammatory diet. Okay? That's what we need to do something about this. Well, summary. Biochemistry stands as a foundational science for understanding the intricate mechanisms of life, health, and human performance. Its historical evolution from philosophical speculation to rigorous scientific discipline has enabled groundbreaking advances in medicine, nutrition, and sports science. Through its applications in clinical diagnostics, pharmacology, genetics, and sports performance, biochemistry continues to shape the future of healthcare and athletic optimization. The integration of biochemistry with cutting-edge fields such as molecular biology, biotechnology, and personalized medicine underscores its ongoing relevance and potential. As research progresses, biochemistry remains as a critical tool for medical innovation, offering new insights into disease mechanisms, therapeutic development, and performance enhancement. Its comprehensive approach to study life at the molecular level ensures that it will continue to be at the forefront of scientific discovery and practical application, improving human health and capabilities. That ends the presentation. Then we can start our Q&A session. It can be held in Spanish. Lo haremos en español para que sea mucho más fácil nuestra comunicación y lo más claro posible. Bien. Estas son mis redes y mi forma de contacto. Ahora sí, estoy abierto a preguntas, respuestas. Ahora sí, me levanto. Me interesa más escucharlos a usted. Sé que ya estudiaron, ya les pusieron exámenes, ya los torturaron con bioquímica. ¿Habrá algún loco que sí le gustó la bioquímica o sí le gusta? A ver, pues algunos loquitos así, sí lo saben. Bien. ¿Tú quieres comentar algo?
No, lo más quería preguntarme si tienen alguna pregunta.
Pues ya vamos. Bien, eso quiero.
En el contexto en el que, por ejemplo, necesitas una emergencia, que necesitas correr, necesitas levantar algo pesado, ¿cómo es que todo este proceso afecta al organismo de una persona?
Estamos hablando sobre entrenamiento sin entrenamiento.
Una persona ordinaria sin entrenamiento.
Vamos a pensar, estás caminando y por ahí te viene cayendo un piano en la escalera.
O sea, una situación que tú estás caminando en la calle
Ajá,
y se cae una persona.
ajá,
Ponemos una persona que es pesada.
ajá.
Y necesitas ayudarla, necesitas
Sí.
levantarla. O sea, ¿cómo está afectada a tu organismo?
¿Cómo reacciona tu organismo?
Ajá.
Ok. Sería el equivalente así. Tú vienes caminando, condiciones normales, ves una situación de estrés. A lo mejor se está cayendo alguien, te viene el piano encima, o peor aún, te viene persiguiendo una víbora de cascabel que viene con patines. Aquí me han dicho que hay víboras de cascabel. No sé si con patines, pero bueno. Tienes una situación en la que necesitas reaccionar rápido. Ocurren dos cosas primero. Una, descarga de adrenalina. ¿Ok? Adrenalina, neurotransmisor. Te va a echar a andar, te va a poner, dicen los españoles, como una moto. Cuando esto ocurre, tu cuerpo va a responder mandándote energía. Necesito energía de una manera rápida y explosiva. Tenemos tres formas de obtener energía, de lo que habíamos platicado ahorita. ¿Sí? Fosfágeno, glucólisis, y teníamos beta oxidación. ¿No? ¿Más o menos? ¿Cuál de las tres es la que responde más rápido? ¿Cuál es la que decíamos que te daba sprints? ¿Cuál? Fosfágeno. Fosfágeno. ¿Ok? El fosfágeno te va a dar energía explosiva y la adrenalina se encarga de esta parte. Te da la explosión. Y esta explosión va a ser intensa. Se han dado casos de mamás en las que se dan cuenta que un coche está a punto de atropellar a su hijo porque se viene de bajada. La mamá no empuja el coche, lo carga para que puedan quitar al niño. ¿Ok? A ese nivel tu cuerpo puede liberar energía vía fosfágeno y vía adrenalina. La adrenalina también te va a liberar ya para un esfuerzo un poco más largo, porque el fosfágeno te va a dar explosiones de 20 segundos, 30 segundos. En 20 segundos no mantengo al coche en lo que sacan un niño. ¿Qué sigue? Noradrenalina va a echar a andar. Primero, adrenalina mueve el fosfágeno y además empieza glicólisis. También es un esfuerzo rápido, toma la glucosa que sí hay en la sangre y de eso se encarga también la adrenalina. La adrenalina levanta concentración de glucosa en sangre. Por eso personas con estrés crónico tienden a diabetes. Es importante. Personas con estrés crónico por la adrenalina tienden a la diabetes, al síndrome metabólico, aunque también hay una parte epigenética. ¿Sí? En una cuestión ambiental sí puede provocar una diabetes. Entonces decían, es que a la abuela le dio diabetes por el susto. No, ya traía una parte predispuesta. Ya venía predispuesta. Pero si el estrés es crónico, porque estos móndrigos, nietos, hijos de toda su gente, nomás la traen con el Jesús en la boca, entonces ese estrés crónico que está liberando adrenalina, sí puede provocar a la larga una diabetes, pero hay una predisposición. ¿Ok? Bien. Volvemos al caso de la mamá que está levantando. Levantan esa explosión de energía, fosfágeno y glicólisis, levanta el peso, lo que se venga encima, por un rato, pero necesito mantenerme más tiempo, ya pasaron 2 minutos, o un poco más. Ahora, la noradrenalina, porque ya ahora es una situación consciente, necesito aguantar. La noradrenalina va a liberar reservas de grasa. ¿Qué ruta metabólica utiliza grasa como fuente de energía? Detoxidación. ¿Ok? Entonces, si te fijas, estoy metiendo las tres cosas juntas. El susto, fosfágeno. Mi esfuerzo fuerte, manteniendo lo más posible. Glicólisis. Me estoy cansando. No, pero tengo que sostenerme. Necesito aguantar. Detoxidación. ¿Sí? ¿Eso es lo que me preguntabas? Perfecto. ¿Qué hace un atleta que está entrenando para un maratón? ¿Cómo aplicaría estas tres formas de obtención de energía en un maratón? ¿Cómo piensas que podría ser?
Pues bueno, que ya tiene el entrenamiento y ya está su organismo acostumbrado a esta actividad.
¿Está entrenado?
Yo diría que, pues, con la beta, ¿no? Porque es más largo, que tiene que se habla bien.
Más o menos. No sé si te has dado cuenta, pero muchas veces los corredores, sobre todo en maratones es más notorio, o los triatlonistas, empiezan a correr y prenden su cronómetro. Pero esos cronómetros también tienen monitores cardíacos. Los monitores cardíacos tienen una función, y es una fórmula, que se llama Carbone. ¿Alguien ha oído de esta fórmula de Carbone? ¿No? Bueno, la fórmula de Carbone es una fórmula que va a relacionar tu frecuencia cardíaca con tu edad y te va a ir marcando en tu monitor cardíaco a qué paso te estás moviendo. ¿Para qué me va a servir eso? Hagan, trae calculadora, sé que lo pueden calcular. Eso pueden calcular cada uno de ustedes. Resten su edad a 220. 220 menos su edad. ¿Ok? Hagan. Les va a dar un número. ¿Sí? En mí está muy fácil. 220 menos 70. 70. Complicado. Bien. Ya obtuvieron su número. Esa es la frecuencia máxima teórica en la que tu cuerpo podría entrenar antes de colapsar. Antes de ¿ok? Bien. Ahora quiero que calculen de ese número que ya obtuvieron, ¿cuánto sería el 70%? Les va a dar otro número, fulano. Y no está muy fácil. O sea, 260, ¿cuál sería las tres cuartas partes? Me va a dar algo así como por 120. ¿Ya tienes su número? ¿Ya apareció por ahí? Bueno, ese número que se encuentra entre el 65 y 75%, serían los márgenes reales, la fórmula, es la frecuencia cardíaca en la que tu cuerpo utiliza grasa como fuente de energía. ¿Ok? Por eso estos monitores tienen unas alarmas. Tú le estableces si quieres que sea entre 65 y 75, tú le marcas a esas fórmulas y se las marcas. Es más o menos el rango, por eso dije 70, para entrar en la mitad. Ok. Por debajo de ese 65, lo voy a decir en español muy castizo, estás haciendo güey y solo estás quemando azúcar. Entonces, si buscabas quemar grasa, a las noticias, no. 65 a 75 es el espacio de tu frecuencia cardíaca y de hecho esos monitores manejan dos alarmas. ¿Sí? Entonces empieza la primera alarma, que es al 65%, tic, 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 tic. Quiere decir que estás entrando en esa frecuencia de quema de grasa. Te empiezas a acelerar porque a lo mejor la música está padre o porque quieres parolear, no sé. Y te pasas de la frecuencia. Ahora empieza otra alarma que dice ti, 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 ti. ¿Qué está pasando? Te está avisando que te estás pasando de esta frecuencia. ¿Qué ocurre después de esa frecuencia? Un proceso que se llama gluconeogénesis. ¿Alguien conoce el término? Vale, ¿qué sí? ¿Sí? Es cuando se convierten las moléculas, aminoácidos generalmente, en glucosa y entran a la glicólisis. ¿Sí? ¿Bueno? Ok. ¿Qué está pasando después del 75%? Tu cuerpo está comiéndose a la masa muscular para mantenerte en paso. ¿Eso me va a servir para ganar músculo? ¿No? Eso es lo que decíamos hace rato como overtraining. ¿Sí? Estás por arriba de lo que realmente tendrías que hacer. Eso te lleva, voy a empezar a mezclar cosas, te lleva a que tu cuerpo empiece a quemar masa muscular para seguirte el paso. Eso te está llevando a un cansancio crónico porque no te vas a recuperar solamente con dormir. Ahora necesito recuperar esas proteínas que acabas de quemar. ¿Sí? Okay. De hecho, en el gimnasio se le llama estás quemando. No sé cuando alguien se está quemando en el gimnasio, es un término que se utiliza. Significa que no va a crecer el músculo y al contrario va para abajo. Muy frecuente en los que hacen CrossFit. ¿Alguien de aquí hace CrossFit o ha hecho CrossFit? ¿No? Niñas tan inteligentes. Es muy frecuente en los CrossFitters que entren en esa situación. Entonces te dicen, no entiendo, estoy haciendo mucho ejercicio, intenso, me voy a ir al Spartan y todo lo demás, competencia. Y no gano músculo, es más, estoy engordando. De las primeras diapositivas, si lograron entender mi inglés, que ya sé, pero bueno, hice el esfuerzo. En las primeras gráficas decía que había tres maneras principales para elevar tu metabolismo basal. Todavía no se dormían en ese momento, ¿eh? Ahora mismo, no todavía no dormían. ¿De cuáles se acuerdan? ¿De qué maneras podríamos acelerar el metabolismo? Una son las hormonas tiroideas. Y no estamos hablando de eso. ¿Qué otras se acuerdan? Hay otras dos.
Todo el cuerpo.
Que decíamos con las tiroides, ajá. Tipo de cuerpo. Ahí teníamos mesomorfo, ectomorfo y endomorfo. Ectomorfo es la persona flaca, flaca, flaca que no logra ganar músculo y es más, lo asustas y baja de peso. Eso nunca ocurre menos en la universidad, ¿verdad? Estamos en exámenes y pierdo peso. No. Ok, eso es un ectomorfo. Es demasiado acelerado. Tenemos al endomorfo, demasiado lento. Dice que hasta el aire lo hace engordar. ¿Okay? Esa puede ser una situación de tiroides, pero bueno. Y hay otro que llamamos mesomorfo. El mesomorfo es el que se supone que tiene un nivel equilibrado de masa muscular. Y decíamos, en esa diapositiva, antes de que cayeran en el desmayo de Morfeo, decíamos que la masa muscular era un activador del metabolismo basal. ¿Sí se acordaban ya? Yo sé que estaban despiertos, él está todavía. ¿Ok? Porque el músculo es de los tejidos que tiene mayor cantidad de mitocondrias. ¿Para qué me sirve una mitocondria? ¿Verdad? Claro, son las fábricas de energía del cuerpo. ¿Ok? Si un tejido tiene mayor cantidad de mitocondrias, y eso lo hace el entrenamiento, decíamos que la mitocondria era flexible y podía multiplicarse, dividirse, agruparse, reciclarse, crecer, hacer formas extrañas, casi hacían figuritas de globo, ¿no? Hay mitocondrias que parecen esto, pero bueno. Con ese entrenamiento yo aumento mitocondrias. Si yo aumento mitocondrias, yo aumento mi capacidad de producir energía. ¿No? Pero también aumento mi necesidad de nutrientes. Aumento mi metabolismo basal. Aquí me sirve, va a ir al gimnasio, va a ir a activar las mitocondrias, ¿verdad? ¿Vas a activar mitocondrias? Nos acabo de dar. Entonces, tu cuerpo se va adaptando con el entrenamiento y esa es una de las cosas que hace, desarrollar masa muscular, aumentar el número o el tamaño de las mitocondrias, aumentar la manera en la que tu cuerpo utiliza qué fuente de energía. ¿Okay? Ahora, ¿qué pasa si tienes todo este entrenamiento, pero no tienes los nutrientes adecuados? Te faltan vitaminas. Vamos a pensar en vitaminas. Yo les dije, cuando estábamos en la universidad, y Marco no me deja mentir, nos hablaron de Marco, mamá de, digo, papá de alguien por aquí. Cuando vimos bioquímica, y lo primero que dije es, vitaminas, perdónalo, hay que, güey. O sea que no, me gustaba la parte de la transcripción del celular, me gustaba la modificación del DNA, los procesos en los que la DNA polimerasa podía regenerar o multiplicar, y biología molecular, o sea, lo apasionante. ¿Cómo vitaminas? ¿Les puedo dejar tarea? Es más, la pueden googlear, a ver si se encuentra. Quiero que me digan ¿Qué vitamina se necesita para fabricar colágeno? Bien. Es correcto. Yo necesito vitamina C para formar colágeno. ¿Bien? La gelatina es una forma de colágeno. De hecho, tenemos en nuestro cuerpo, depende de quién lo tome, pero tenemos 13 tipos diferentes de colágeno. Uno forma el cabello, otro forma las uñas, otro forma los manos, los ligamentos, la nariz, la oreja. O sea, tenemos 13 tipos. Y si eres un feto, 19. Y todos son formas parecidas a la gelatina. ¿Sabes cómo se obtiene la gelatina? Cualquier tecnólogo de alimentos podría explicar eso. ¿De dónde sacamos la gelatina? Pues se hierve. Sí. Nadie de ustedes ha hecho manitas de puerco. Nadie ha probado las tostadas de papa. Ok. Nunca les ha tocado ponerse a lavar la olla o donde hayan estado las manitas. ¿Qué hay cuando estás lavando, cuando vas a lavar la olla o sea, el recipiente? ¿Qué es lo que hay en el fondo? Hay una cosa ahí medio extraña, ¿no? Y no son las manitas. No, nadie de ustedes lava los trastes. Sí. En el fondo de esa olla te vas a encontrar una cosa que esa cosa que la tienes y esa es la cara, esa es la que luego te venden carísima para los amigos. Y la podías haber hecho en tu casa. Pero bueno, hay una reacción para formar colágeno que se llama hidroxilación de la colina. Y eso requiere vitamina C. ¿Para qué me puede servir? ¿Para qué me sirve saber que puedes formar colágeno a partir de la vitamina, con la vitamina C? ¿Qué se te ocurre? Recuperación. Sí, ¿verdad? Pulmones, ligamentos. Y vimos que tenemos 13 tipos diferentes de colágeno. Te lo voy a llevar a otro nivel. ¿Sí? Si yo te dijera que puedo encapsular un tumor en una cápsula de colágeno y así evitar que se multiplique e invada el cuerpo. ¿Ves? Investígate el protocolo COP, el encapsulamiento del tumor. Es algo que utiliza. Cáncer de mama, cáncer de próstata, cáncer de pulmón, cáncer de riñón, cáncer de hueso. Son indígenas. Ahora dime qué hueva con la energía. No. Si yo te dijera que incrementando la masa muscular, incrementando las mitocondrias en un atleta paralímpico, recién operado del hombro, y que creyó que su carrera estaba terminada. Si yo te dijera que en 10 meses logramos recuperar el hombro, de correr silla de ruedas, y tenerlo en condiciones como para poder competir en las Olimpiadas de Río, sí, de Río, y traerse 2 medallas, un bronce y una plata, cuando creía que su carrera estaba terminada. ¿Ves? César Navarro, mi paciente, y extracto. Y el desgraciado no me dejó correr con su silla de ruedas en Magdalena Michoacán. Yes. Sí. Si yo te dijera que tenemos casos de pacientes con síndrome metabólico programado por parte de sus papás, dos, porque hombres y mujeres transmiten esas partes genéticas, y que yo he podido mantener por 20 años la diabetes o el síndrome metabólico controlado a pesar de las condiciones. ¿Ves? No, tengo familias así. Familias. Si yo te dijera que tengo pacientes que tuvieron un accidente en la moto, voltearon con la cabeza en una pipa, y que hemos podido sacarlo adelante, aumentando neuroregeneración con ciertos suplementos y tenerlo nuevamente en la universidad. Y lo que- ¿ok? Vitaminas, minerales, aminoácidos. Ay, Si yo te dijera, no hemos hablado casi de aminoácidos, si yo te dijera, sí les dije que la serotonina te aumentaba, ¿no? Y por eso estaban así todos los mismos. Oye, bueno, si yo les dijera que puedo mantener controlado, tranquilo, a un paciente esquizofrénico, ¿eh? Con solo aminoácidos, bueno, no sé, cuidando que no haya alergias alimentarias. ¿A dónde quiero llegar? Y esa es mi conclusión, lo que quiero cerrar ya, porque tiempo es tiempo. Me robé 5 minutos, perdón. Cuando yo estaba en la universidad, bioquímica, qué bueno. O sea, qué loco, eso es lo apasionante. Ahora te puedo decir que cuando lo usas con el conocimiento aplicado, ya te platiqué varios de estas mis experiencias. Si yo te dijera que puedes ayudar y cambiar, transformar, ayudar a cambiar vidas, familias. Puedes mantener a raya por más de 10 años un tumor que aparentemente iba a consumir una persona en 6 meses. Si te dijera que puedes mantener pacientes con síndrome de desgaste, que es el síndrome de desgaste, personas que tenían una sentencia de muerte, literal, así me llegaron. Y decir que después de 10 años, ahí sigue, e inclusive después del síndrome de desgaste, y le sigue, ¿no? Esa parte está interesante. Ahora están compitiendo en fisicoculturismo. ¿Me lo crees? Número 3. Ya revisar. Definición de síndrome de desgaste es la pérdida de más del 10% del peso magro de un paciente en menos de 6 meses y sin estar haciendo ejercicio ni dieta. Está asociado a diabetes, a cáncer, hepatitis, una serie de enfermedades crónicas. Se están chupando, llegando a huesitos. Los recuperamos y ahora están compitiendo. Físico culturismo. Sí, en categoría fija, porque está el desayuno. Pero sí, 2 veces traje de baño y dice, ¿dónde está el descanso? ¿A qué quiere llegar? Espero, ojalá, que alguno tenga la curiosidad de ir un poco más allá del libro de lectura. ¿Cuál están llevando? ¿De ninja? No.
Generalmente sí.
¿De ninja? ¿El coso azul? Es bueno para dormir. Yo tengo ese. Bueno, no se queden solamente en el libro. Está padre, el libro me gusta. Siempre tuve, Marco, no me dejará mentir, siempre tuve la inquietud de, ¿y esto cómo diablos se come? ¿Para qué me sirve? Siempre veamos más allá. Casi no hablamos, aunque lo mencionamos, de los omega 3 y omega 6. Y mencionamos un poquito. Les pongo una tarea, que eso puede ser para el resto del tiempo. Si yo controlo inflamación celular, yo controlo la epigenética. ¿Sí? Si yo controlo la inflamación celular, yo puedo controlar la expresión epigenética. Tiene tarea. Alguno responderá, espero que sea más de uno, y el que lo haga, bienvenido a mi mundo. ¿Ok? Gracias por estar aquí.
Bueno, pues concluimos por el día de hoy. La plática siguiente se canceló porque el esposo de la invitada tuvo un problema. Muchas gracias.
Agradecido esto.
Los que están